Hello, and let us discuss cleavage, division and implantation today. After fertilization, the zygote starts dividing repeatedly. This repeated mitotic division of the zygote is cleavage division. The result of this cleavage division is there is increase in cell number and at the same time there is decrease in cell size. Such newly formed embryonic cells as a result of cleavage are known as blastomeres. So upon the first division you get 2 cells, upon the second division you get 4 cells, upon third you get 8 cells. After the third cleavage, the blastomeres align tightly by maximizing areas of their contact to form a compact ball of cells. This process is known as compaction. The compaction is mediated by cell adhesion glycoproteins, and this compaction is necessary for the blastomeres to decide early on which of the blastomeres are going to participate in formation of tissues and organs of the embryo and which ones are going to contribute in formation of the nutrition system of the embryo. By day 3 after the fertilization, the compacted cells divide to form a 32 to 36 cell clump. At the level of 16 cell itself, it is known as morula. And this morula, by the time it enters the uterine cavity, day 4 has arrived. And this morula is showing 2 clear-cut areas, the inner cell mass and the outer cell mass. As it enters the uterine cavity, gradually small spaces start appearing between the inner and the outer cell mass, and these small spaces coalesce to form a cavity which gets filled with fluid, and this cavity, newly formed one, is called the blastocystic cavity. The embryo now will be known as a blastocyst. The inner cell mass is going to form the embryoblast, that is tissues of the embryo, and the outer cell mass is going to form the nutrition system of the embryo, that will be trophoblast. A noteworthy point here is the zona pellucida, which was present all along till now, degenerates by 4 to 6 days after fertilization, and it is only after the degeneration of the zona pellucida that there is any chance for this conceptus to actually implant into the uterus. Usually by day 6 we witness the process of implantation. The blastocyst, which is hatched out of zona pellucida, attaches to the endometrium at the embryonic pole. So the blastocyst will have 2 poles, an embryonic pole which is in close proximity with the embryoblast, and at the exactly opposite pole is the abembryonic pole. So the blastocyst attaches to the endometrium at the embryonic pole. The common site for implantation is upper part of posterior wall of the uterine cavity. By then, when the embryo starts attaching to the wall of the uterine cavity, the trophoblast has started differentiating into 2 layers, a syncytial trophoblast and a cytotrophoblast. So if first week of embryonic life needs to be summarized, we can say by day 1 it is fertilization, by day 2 we get 2 cell stage, day 3 is the day when we get morula, by day 4 embryo enters uterine cavity and forms early blastocyst, by day 5 we get a late blastocyst and the zona pellucida disappears by then, and on day 6 the process of implantation actually begins. So the implantation begins on day 6 of life. The blastocyst has an outer layer of cells, trophoblast, and the inner cell, the embryoblast. The uterine endometrium by then is in secretory phase. The blastocyst is partially embedded in the endometrium by day 7, and this embedding of blastocyst into the endometrium happens due to phagocytic and burrowing properties of the syncytial trophoblast. It is interesting to note that the cytotrophoblast is the inner layer. It consists of uninucleated cells and the cell margins are distinct, whereas the syncytial trophoblast is the outer zone. It consists of multinucleated protoplasmic mass where the cell margins are indistinct. As far as the properties of the cytotrophoblast and syncytial trophoblast are concerned, the syncytial trophoblast is an aggressive layer. It can eat up and digest any cells or cell lining it comes in contact with, and precisely that is the reason why syncytial trophoblast is at the advancing front of implantation, while it is trying to establish itself into the wall of the endometrium, that is at the level of uterine endometrium. In addition to being aggressive, the syncytial trophoblast also secretes a hormone, the human chorionic gonadotropin, also known as HCG. The HCG is responsible in maintaining the corpus luteum of pregnancy. The progesterone that is released from corpus luteum is a pregnancy maintaining hormone, and if this conceptus has to stand a chance of developing all the way till the period of maturity and eventually reaching the stage of parturition, it needs to have mechanisms that can maintain high levels of progesterone. So early on, the HCG is the one that maintains corpus luteum so that the corpus luteum can continue to pour in required amounts of progesterone in the bloodstream. Radioimmunoassays can detect this HCG in maternal blood as early as the second week of gestation, and thus HCG forms the basis for early pregnancy test. The syncytial trophoblast has the capacity to erode endometrial cells as well as the blood vessels that come in its path. So as of now, in the trophoblast we got two layers. The outer layer is syncytial trophoblast, which grows aggressively and can eat up whatever comes in its way, and the inner layer is cytotrophoblast, which is comparatively docile, so to say. The trophoblast continues to develop by the time we see the day 9 has arrived, and by then, because of the aggressive dividing properties of the syncytial trophoblast, small spaces start appearing in the syncytium. We call them vacuoles near the embryonic pole, and once you start seeing these vacuoles coming at more than one places, this particular stage of trophoblast is called the lacuna stage, and we are about to witness how the nutrition mechanism of the embryo, that is establishment of placenta, is going to take place. In that, we can say that the initial step is a lacuna stage where spaces start appearing in the syncytial trophoblast. By day 11 and 12, the lacuna spaces in the syncytial trophoblast intercommunicate so that the lacuna networks can form, and syncytial trophoblast penetrates deeper into the uterine stroma, and it is ready to come in contact with maternal capillaries or maternal sinusoids as they are known as, and those will be eroded. Once the maternal sinusoids are eroded, the lacunae communicate with these maternal sinusoids, and now the maternal blood starts flowing into the lacunar spaces or the lacunar network of the trophoblast. That is, maternal blood flows into the trophoblastic system, and at this point of time it can be said that the uteroplacental circulation is established. By this time of day 11 and 12, the blastocyst is embedded deep in the endometrium, and the surface rent that was created because of the burrowing journey of the blastocyst, the surface rent is almost completely covered. The epithelium covers the defect, and there is formation of a small endometrial plug. The blastocyst produces mild protrusion into the wall of the cavity by now. If these are the changes that were happening in the trophoblast, what are the changes that are happening in the embryoblast? Let us talk about them. The embryoblast, which is represented by inner cell mass situated at the embryonic pole, starts separating into tall cells towards the embryonic pole and comparatively flatter cells towards the abembryonic. So the layer of tall columnar cells, it is the one which is seen towards the embryonic pole, is the epiblast, and a layer of small cuboidal cells which is adjacent to this epiblast, the cells there, the layer there is termed as hypoblast. So embryoblast now differentiates into two layers, epiblast and hypoblast. This is day 8. The hypoblast and the epiblast together form a flat bilaminar germ disc. We call it embryonic disc, and close to the epiblast a small cavity appears, and this cavity will eventually enlarge to give rise to amniotic cavity. The epiblast cells adjacent to the cytotrophoblast are called amnioblast. The epiblast and amnioblast together line the amniotic cavity, and this cavity comes to contain amniotic fluid. By day 9, at the abembryonic pole, flattened cells originating from hypoblast form a thin membrane known as exocoelomic membrane or Husser's membrane. Along with the hypoblast, this newly formed Husser's or exocoelomic membrane lines a cavity that is called exocoelomic cavity or primary yolk sac. So now, inside of the trophoblastic shell, we have the embryo being represented by bilaminar germ disc, epiblast and hypoblast, and we see that the embryonic disc, which is bilaminar, is sandwiched between two cavities. Towards the embryonic pole of the embryo, we see the amniotic cavity, and towards the abembryonic pole of this bilaminar germ disc, we see exocoelomic cavity or the yolk sac. By day 10, the blastocyst is deeply embedded in the endometrium and the penetration defect is closed by a fibrin coagulum. By days 11 and 12, new cells derived from the yolk sac appear between cytotrophoblast and the exocoelomic cavity, and they form a new layer called extraembryonic mesoderm, also known as the primary mesoderm. Soon, cavities develop in this extraembryonic mesoderm, and slowly they coalesce to form a horseshoe-shaped cavity that appears horseshoe-shaped in a section. This newly formed cavity is called extraembryonic coelom or the chorionic cavity. The chorionic cavity surrounds the primitive yolk sac and amniotic cavity, and this cavity does not extend into the area where the germ disc is connected to the trophoblast. So this part of extraembryonic mesoderm that suspends the developing embryo, amniotic cavity and yolk sac in the chorionic cavity is the connecting stalk. By the formation of this chorionic sac, the extraembryonic mesoderm is subdivided into a parietal layer and a visceral layer. The parietal layer is the one that will be in contact with the cytotrophoblast, and the visceral layer that will be in contact with the yolk sac and the amniotic cavity. So this parietal layer is also termed as extraembryonic somatopleuric mesoderm. It lines the cytotrophoblast and amnion and is also called as chorionic plate, whereas the extraembryonic splanchnopluric mesoderm, the visceral one, it lines the yolk sac. The bilaminar germ disc grows slowly in comparison to the trophoblast. Meanwhile, when these changes are happening in the trophoblast and the embryoblast, the endometrium of the uterine cavity also shows certain changes. The cells in the endometrial stroma by day 11 and 12 undergo decidual reaction, in which the cells become polyhedral and get filled with glycogen and lipids, and the cells also become edematous. Some of them might also undergo apoptosis, and because of the secretion and presence of this prostacyclin, this edematous endometrium, which is now termed as decidua due to the decidual reaction, creates an immunologically privileged environment for implantation, because the newly formed conceptus, that is embryo, is genetically different from that of the mother. So if it comes in contact with the maternal circulation and the white blood cells in particular, the white blood cell might seem to find this embryo as a foreign body and would probably march an immune reaction. In order to prevent that, immunologically privileged environment needs to be created, and that exactly is done by the decidual reaction of the endometrium. Now by day 13 arrive, the surface defect of the endometrium is completely healed, and at that particular time bleeding may sometimes occur at the site of implantation, which can very well be confused with normal menstrual bleeding, and that might be a source of the wrong dates as they are given by the mother during the time of antenatal checkup as to the last menstrual period. The dates calculation goes wrong because of this particular observation. The hypoblast produces more cells and they migrate along the inside of the exocoelomic cavity. These cells proliferate and a new smaller cavity is formed within the exocoelomic cavity. This newly formed cavity now by day 13 is called a secondary yolk sac or a definitive yolk sac. As the secondary yolk sac is being formed, portions of the exocoelomic cavity are pinched off to form exocoelomic cysts, and at this point of time these cysts lie in the chorionic cavity. And now come day 13 and 14 and the trophoblast forms primary chorionic villi. Remember, the trophoblast arrangement is in the form that outer layer is syncytial trophoblast, inner layer is cytotrophoblast. Now these cells of cytotrophoblast proliferate. They penetrate into the syncytial trophoblast, and these cellular columns which are formed as a result of this penetration of cytotrophoblast cells into the syncytial trophoblast, the cellular columns of cytotrophoblast comes to get covered by syncytium, and this is the structure of a primary chorionic villus. Another important event that happens on day 14 within the embryoblast is the embryoblast at that stage is present in the form of a flat bilaminar germ disc. Now out of the two layers that we have, epiblast and hypoblast, the hypoblast cells in a particular area, the hypoblast cells become columnar. Elsewhere they are cuboidal, but at one particular area they become columnar. And they not only get columnar, but they tightly get attached to the overlying epiblast cells. And this circular thickened formed area now is known as a precaudal plate. This precaudal plate indicates the future site of the mouth of the developing embryo, and once that happens, the craniocaudal axis is established, and this happens on day 14. In whatever developmental changes that have happened in the embryo, the trophoblast and the endometrium, a couple of clinical correlates needs to be kept in mind. Number one is ectopic pregnancy. The normal site of pregnancy implantation is in the posterior wall of the cavity of the uterus, but if it doesn't happen there, it can happen in the fallopian tube. It might happen on the surface of the ovary. It might happen in the cervix of the uterus, or it can happen onto the any surrounding abdominal viscera. Now all those areas apart from the upper part of posterior wall of the cavity of uterus, all other pregnancies probably will not reach their final logical development and conclusion, and they might probably get aborted at one point of time or the other, or may even endanger the life of the mother. Other important clinical correlate is a hydatiform mole. Now this is an abnormal blastocyst. There the trophoblast develops to form membranes, and there is little or no embryonic tissue. Placenta contains small cyst, and this is a potentially malignant condition, and that can very well lead to a cancer called the choriocarcinoma. So the early 2 weeks of life are the week of twos, where trophoblast differentiates into two layers, embryoblast differentiates into two layers, 2 cavities are formed inside the embryo. Extra embryonic mesoderm forms 2 layers. Well, that's the initial 2 weeks for you. Happy learning.